Hello everyone, welcome to today's live broadcast, Filtration and Dissolution Testing. I'm Ali Matheson, Editor-in-Chief of LCGC Europe, and I will be your moderator for today's event. We are very pleased to bring you this webcast presented by LCGC and sponsored by Merck KGaA Darmstadt, Germany. Millipore Sigma is the US life science business of Merck KGaA Darmstadt, Germany, with 19,000 employees and 72 manufacturing sites worldwide. Millipore Sigma's portfolio spans more than 300,000 products, enabling scientific discovery. Millipore Sigma has customers in life science companies, university and government institutions, hospitals and industry. The company is committed to solving the toughest problems in life science by collaborating with the global scientific community. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. I would now like to introduce today's speaker. We are very pleased to be joined today by Vivek Joshi, PhD. As an Applications and Market Adoption Manager at Millipore Sigma, Vivek Joshi develops new products and applications for products spanning analytical, molecular and protein science and cell culture workflows. Prior to joining Millipore Sigma, Dr. Joshi worked on high-throughput LC-MS applications at Arquil. Dr. Joshi's doctoral research focused on molecular imprinted polymers, and he completed his postdoctoral studies of molecular imprinted polymers and capillary electrochromatography. Recently, Dr. Joshi presented a sample preparation module in the drug dissolution training offered by the United States Pharmacopoeia. And thank you very much for joining us today, Dr. Joshi. If you start the presentation, that would be great.
The life science business of Merck KGaA Darmstadt, Germany operates as Millipore Sigma in the US and Canada. Today we are going to talk about filtration in dissolution testing. My agenda is going to be, I'll be talking about importance of dissolution testing, followed by why is filtration critical in dissolution testing, some of the membrane characteristics, throughput in filtration, and finally membrane characteristics that impact data. Before we get into the workflows that you are well aware of, let's talk a little bit about scope of pharma QC. There are a variety of tests carried out in pharma QC, many of which are mandatory and required by FDA. A lot of these tests involve filtration as a means of sample preparation, and sample preparation concerns which are similar to drug dissolution testing. To give you a perspective on numbers, more than 25 to 35 million drug dissolution tests are performed every year. Other than dissolution tests, various pharma QC tests like assay, content uniformity, stability and shelf life, as well as physical and mechanical testing of dosage forms also require filtration for sample clarification. Dissolution testing is performed throughout the drug development cycle, but the goals of dissolution change as the drug moves through the development. During early drug discovery stage, when new chemical entities are being discovered against new targets, no dissolution test is carried out. But as the drug moves into development, formulation development begins, and goal of dissolution test at this point is to help formulation scientists with how the formulation impacts dissolution profile. This is even more critical for extended or modified release formulations as they need to release drugs in a certain fashion. Once the drug moves to approval stage, dissolution test data is an integral part of NDA documentation and FDA requirement. As the drug moves into production, dissolution test becomes a QC test which helps understand effects of process and manufacturing change on dissolution profile. For instance, What happens when a new lot of excipient is used in the formulation, or what happens to dissolution profile when a new supplier for a certain excipient needs to be selected. Dissolution test helps pass or fail a drug lot. Finally, as the drug moves into the generic arena, dissolution test is one test which helps prove bioequivalence of this generic drug with the patent-protected innovative drug on the market. During all these stages, scientists are always looking for ways to find out a correlation between the in vitro dissolution test and in vivo concentration profile, or IVIVC. Now let's take a look at the dissolution testing workflow. Various steps of drug dissolution workflow are presented here in this slide. Some of the concerns at these various steps are also listed alongside. The concerns that are listed here are mainly associated with sample preparation and quality of components of media and mobile phase if HPLC analysis is carried out downstream. For example, during media preparation, deaeration of dissolution medium is a key concern, as presence of air or oxygen dissolved in dissolution medium affects dissolution profile. One such well-known example is the prednisone tablet, which is a USB calibrated tablet used for calibrating dissolution baths. And presence of oxygen in dissolved medium can affect rate of dissolution. Vacuum filtration is one of the well-known methods for deaeration of dissolution media. For oral dosage forms, most commonly used dissolution apparatus is either apparatus 1, basket type, or apparatus 2, paddle type. But some other apparatus may be used for different dosage forms, such as patches or repositories. Based on the dosage form, many a times automatic or manual sample collection is selected. Commonly for extended release formulations, automated sample collection is preferred over manual sample collection. Filtration is the key sample step that is involved during sample collection. Sample analysis is commonly done either using UV-vis spectrophotometry or HPLC coupled with UV or some other detector. These days, HPLC is becoming the most common way of analyzing sample after dissolution test, simply because many a times you have very low concentration of analyte in the dissolution medium. Sometimes there are multiple components which can interfere with UV-vis spectrophotometric analysis. Finally, the data is analyzed to generate dissolution profile to compare it with the existing dissolution profile. So now that we talked about dissolution test and its importance, let's talk about why filtration is critical in dissolution testing. Filtration stops dissolution process. Filtration of the sample media is required to ensure that most representative sample is analyzed. Filtration is required here as it stops the dissolution process. Dissolution medium contains both the dissolved and undissolved inert ingredient as well as your active component. The filtrate, when post-filtration, will contain the dissolved ingredients, a more representative sample at that time point. If any solid particles are allowed to pass the analysis step, inaccurate data could result either from particle interference or downstream dissolution of drug which was still undissolved. And that's why filtration is important in dissolution testing. Commonly, there are two means of filtration following a dissolution test. One using syringe filters and one using Fritz or cannula filters. If you take a look at a typical USP dissolution protocol, it always recommends that the analysis be done on the filtered portion of solution under the test. Now let's compare the two dissolution methods that I highlighted in the previous slide. Syringe filter versus Fritz filter. Pore sizes of Fritz are typically large, anywhere from 10 micron to 70 micron, as opposed to syringe filters, which are generally between 0.1 to 1 micron. Very commonly, a 20 to 30 micron Fritz filter is used in dissolution. What that means is that the sample will need to be further filtered using a syringe filter for downstream HPLC analysis. Even if the analysis was to be done by UV-vis spectrophotometry, some interference can be observed due to presence of particulates. So the typical pore size difference between syringe filters and frit filters can be substantial when it comes to downstream analysis. Another important difference between frits and syringe filters is that their pore size rating is an average pore size, which implies that the filtration performance of a 30 or a 40 or a 50 micron frit can be very similar. On the other hand, the pore size distribution for a syringe filter is very narrow because the pore size rating for a syringe filter is provided by the highest pore size. And what that means is a 0.45 micron syringe filter will have all the pores smaller or equal to 0.45 micron. Other than these two key differences between the frit and syringe filter, they have very similar materials of constructions. They can be used in backwashing. Sometimes they can also be reused. And both frit and syringe filters are compatible with automated and manual methods. Overall, what we have observed is that syringe filters are better suited for particle removal compared to frit filters. And especially with HPLC becoming the dominant downstream analytical technique, even if frit filters were used as part of automated solution, further downstream filtration using syringe filters is still required. Some of the key concerns that a customer has when they think about filtration are listed here in this table. For example, a customer may be concerned about a filter which allows for filtration directly into HPLC vial because that can prevent multiple transfer steps. Sometimes, if customers are switching from HPLC analysis to UHPLC analysis, they are concerned whether the same pore size syringe filter that they were using previously is suitable for UHPLC analysis. A lot of times customers still use frit filters as part of their automated solution. And they are aware that frits have much broader pore size distribution compared to a syringe filter, and hence they would like to get frits which have much narrower pore size distribution. Filters also show variable drug binding and extractables, so a filter without either of those would be really great. If you condense all these questions into one key question that the customers have in their mind, that question is, are we using the right filter if I chose a filter that worked well last time with compound X? Now that researchers are working with a different compound with different physico-chemical properties, the same filter that worked previously may or may not work in their analysis. So even though customers have these concerns about how a filter is selected, when you ask them how they select a filter normally, what comes up as the top answer is something that is present in the drawer or stockroom. I have done these seminars at a number of pharmaceutical customers, and most common answer that I get is, oh, I use the yellow filter that was in my drawer, or I use the red or blue or green filter. Something that comes up over and over again is the one that worked last time. And again, it may work this time as well, but if it doesn't work, we don't know why it does not work. Some customers who are working on automated filter changing stations, they need a filter which is automation compatible. Sometimes the equipment manufacturers recommend certain filters. Typically for QC labs, the SOPs or the USP protocol dictates which filter needs to be used. But the important criteria, by pore size, by membrane material, it must have pre-filter and chemical compatibility, generally don't come in people's mind when they are selecting a filter. And as I said, in 80% of the cases, the filter that was chosen will work, but if it doesn't work, then we have no reason to know why it did not work. So now let's talk about some of the key membrane characteristics which impact the filtration performance. Previously, I talked about pore size of membrane filters being 0.45 micron or 0.2 micron or 0.1 micron. So how the heck do we measure these pore sizes? We can definitely not measure them by a ruler or a micrometer because these are too small pores to measure. One of the most commonly used methods for measuring pore size of a membrane filter or a syringe filter is by a test known as bubble point. Bubble bubble point is a pressure that is needed to allow flow of air through a wetted membrane. This pressure can be correlated to pore size. Since the largest pores on a filter will have the least resistance for airflow through the filter, bubble point measure typically gives an answer about largest pore in membrane filter. The second means for measuring pore size of a membrane is by bacterial challenge. This test is more important for sterile grade filters where a bacterial suspension of certain size bacteria is allowed to pass through a filter and the bacterial particles on the downstream side are evaluated. Both the bubble point and bacterial challenge are test methods which have been standardized for a number of years by millipore sigma. But both of these are indirect methods of measuring pore sizes. Some of the direct measures of measuring pore size are scanning electron microscopy and porosimetry. But these are generally used in R&D for pore size measurement. Here, there are various scanning electron microscopes or micrograph pictures of various membranes. And let's talk about why the pore size and the pore structure looks very different from one another. These are scanning electron micrograph images of various 0.45 micron membranes. As can be seen that even though these have the same average pore size, their pore morphology and pore structure looks very different. And the reason for this difference is the way in which these membranes are manufactured. For instance, the PVDF, the PES, and nylon membrane are made by phase separation using a solvent and a non-solvent combination. Or in polymer synthesis term, these are called as solvent cast membrane. A mixed cellulose ester or MCE membrane is also manufactured by a phase separation method where air is used as a non-solvent. The polycarbonate membrane presented at the left-hand corner is manufactured by a track etching process, whereby a sheet of polycarbonate is irradiated followed by chemical etching. This is one of the very few membranes which have truly cylindrical pores. On the top right-hand side, we have polytetrafluoroethylene or PTFE membrane, where the pores look stretched in one particular dimension. And that is because this particular membrane is made by stretching of a PTFE tape similar to a plumber's tape. And the direction of stretch will introduce the pores, and they are elongated in that certain direction. Manufacturing process overall determines the pore structure and morphology of the membranes. Many times the porosity of these membranes also differ. Percent porosity essentially indicates amount of air these membranes hold. Except for polycarbonate membranes shown here, all the other membranes generally have a porosity in the range of 70 to 80%. The polycarbonate membrane, on the other hand, only have about 15 to 20% porosity, which can impact its filtration performance. Sometimes the membranes might have the same pore size, but their pore structure across the thickness of the membrane can vary. The structure shown at the top is a symmetric Durapore or a PVDF membrane, where the pore size and pore structure is the same at top and the bottom. Whereas the bottom panel shows an asymmetric millipore express plus membrane, which is a polyether sulfone membrane, where the top surface has larger pore size compared to the smaller pore size at the bottom. And what that funnel-like structure does is it improves the filtration performance of the membrane by keeping the retention of particles at the same level. In HPLC analysis, which is very common after dissolution testing, retention of particles by syringe filters is critical. In this example, we are showing retention behavior of 4 different syringe filters from 2 different vendors. The PTFE, PVDF, nylon, and PES syringe filters from vendor A, as well as the polypropylene, PVDF, nylon, and PES syringe filter from vendor B. As you can clearly see, for nylon and the PES membranes, both the membranes show similar retention behavior, that is greater than 95% retention of particles. To give you a background, this experiment was done using 0.2 micron syringe filters, and what we did was we filtered 0.3 micron latex suspensions through these syringe filters. As I said earlier, the largest pore size in a membrane is the rated pore size of a syringe filter. All these 0.2 micron syringe filters should be able to retain all the 0.3 micron particles, which is observed for both the nylon and PES membranes. But with PTFE compared versus polypropylene and PVDF compared across the two vendors, both the PTFE and PVDF from vendor A are able to retain quantitative particles, whereas the polypropylene or the PVDF membrane from vendor B only retains about 80 to 85% of the particles, letting about 15 to 20% of the particles go through. And that's why it is important to know the differences between different membranes and different vendors. The same example now is manifested by an increase in back pressure of an HPLC or a UHPLC system. In this example, what we did was we filtered a mobile phase consisting of 50% acetonitrile and 50% water using the same two membranes as shown in the previous slide, the PTFE from vendor A and the polypropylene from vendor B. This mobile phase was then run on a Waters Acuity UPLC system for a period of 10 hours, and the back pressure was measured every certain amount of time. As you can clearly see, the membrane which allows for complete retention of particles, the back pressure does not go up at all over the 10-hour period. On the other hand, the polypropylene membrane filter, which did allow some amount of particles to go through, these particles get collected at the head of the column, thereby leading to an increase in back pressure in this UPLC system by as much as 1500 to 2000 psi. So now imagine if this UPLC system was also running the samples that were filtered through the same polypropylene syringe filter, you can imagine how much increase in back pressure this can lead to. And in some times, this UHPLC system can shut down completely altogether. So it is important to understand the differences between membranes and syringe filters between different vendors. Another key important characteristics of membranes or syringe filters is their chemical compatibility. A syringe filter has a housing, which is generally made out of HDPE and polypropylene, and both these materials are generally compatible with a wide range of chemicals, including acids, bases, surfactants, and organic solvents. Of the membrane materials, the PTFE membrane has the highest chemical compatibility, followed by PVDF, nylon, and PES, as well as regenerated cellulose. You might wonder here how the PTFE membrane is able to filter aqueous solutions. The PTFE membrane that we are talking about is Millipore Sigma's hydrophilic PTFE membrane. A PTFE or Teflon membrane has very high chemical compatibility and is compatible with a wide range of chemicals, but it is also hydrophobic in nature. What we do within Millipore Sigma is create a hydrophilization treatment for this PTFE membrane, which retains the chemical compatibility behavior of the original hydrophobic material, but allows filtration of various aqueous solutions. So this hydrophilic PTFE membrane has the highest chemical compatibility of all the various membranes studied. Now that we have looked at some of the characteristics of membranes, let's talk about throughput. The first and foremost question when we are filtering a sample to ask is how much volume you are filtering, because that can help you choose the right size filter for the application. Use of large filter for small volume filtration can lead to loss of sample through hold-up volume, sometimes difficulty in filtration, and also lead to some extractables and non-specific binding concern. Specifically for dissolution testing application, where typical sample volume filtered is about 10 to 20 ml, a 25 or a 33 millimeter syringe filter is the right size filter for this application. If there is a concern about analyte binding or extractables contaminating the sample, a 13 millimeter syringe filter can also be used for dissolution testing application. simply because a 13mm syringe filter has much lower surface area, thereby showing lower analyte binding as well as lower extractable concerns. In general, the right size filter will improve throughput, reduce sample loss due to holdup, and minimize extractable and analyte binding. Sometimes improving throughput can be using a vacuum-based filtration system which allows for filtration of up to 8 samples at a time. The example shown here is the Millipore Sigma Simplicity G2 system. This is a vacuum-based sample filtration prior to HPLC or UHPLC analysis. This system lets filtration directly into an HPLC vial. And the benefit of this system is that you can filter anywhere from 1 sample to 8 samples at the same time. The next challenge that comes in filtration is when we are trying to filter high particle suspensions. So typically, how do people filter difficult samples, like samples which contain large amount of particles? Because a membrane syringe filter can easily clog when you have large amount of particles, customers use multiple syringe filters to get the desired volume. Sometimes they have to use a coarse pre-filter followed by final membrane filtration. Or you can ask the strongest person in the lab to filter the sample. That doesn't make you a lot of friends in the long run. A better solution would be a syringe filter that contains multiple layers of pre-filters and a final 0.45 and a 0.2 micron membrane that will improve throughput. The data shown on the right-hand panel shows filtration of 3 different solutions using an HPF or high particle filtration solution syringe filter from Millipore Sigma compared against a membrane-only PVDF syringe filter. And as you can see, up to 6 to 10 times higher filtration volume is possible using a pre-filter containing syringe filter. Many times, in order to improve filtration performance and improve throughput, customers resort to automation. Some of the key challenges for automation is that you need an automation-compatible filter, as well as confirming that the data is not influenced by auto-sampling. If you know that the formulation in the downstream process is going to be tested using an automated filter changing station, it's better to start developing the method on an automated system, because that way you can validate a syringe filter which is compatible for automation systems, and this method can be transferred to the QC lab downstream. Millipore Sigma offers a variety of solutions for filtration of sample using an automated system or automated filter changing stations. For filtration of viscous solutions, that is, the solutions which contain dissolved impurities, sometimes an ultrafiltration membrane-based filter such as an Amicon Ultra can be used for dissolved impurities. Many of the extended release formulations these days contain polymeric excipients which, when dissolved, increase viscosity of the solution. These formulations may not contain large amount of particles, so an HPF or a pre-filter version of the filter does not help as much. An ultrafiltration membrane-based device allows for removal of dissolved impurities based on their molecular weight. And generally, the molecular weight of APIs is much smaller than the polymeric excipients that are used in this application. A 10 or a 30 kilodalton syringe filter with ultrafiltration membrane can easily be allowed for filtration of these solutions. Generally, these devices are used for protein concentration or desalting, but in pharmaceutical application, they can be used for removal of polymeric excipients. In the final section of this presentation, I'm going to talk about some of the key membrane properties that impact the data. We already talked about chemical compatibility of housing and the membranes. And we saw that TFE has highest chemical compatibility, followed by PVDF and nylon, followed by polyether sulfone and regenerated cellulose. The two other key concerns of scientists working in dissolution testing is drug binding, as well as extractables. Drug binding changes the amount of analyte that is present in the final filtered solution, thereby leading to incorrect quantitation. Extractables are low or medium molecular weight impurities that get leached out from the syringe filter or any other surface that comes in contact with the sample, and those can impact the downstream HPLC analysis. So let's talk about drug recovery, which is dependent on membrane and analyte. In the left-hand panel, here we are showing drug dissolution testing results for 4 different drugs of interest. These formulations were containing ranitidine, acetaminophen, ibuprofen, or loratadine as the analytes of interest. A typical dissolution test using standard USP protocols were performed on these formulations, and after the dissolution test was complete, the samples were filtered through either a nylon syringe filter or a hydrophilic PTFE syringe filter. And various fractions of the filtrate were collected, starting from the first mil of filtrate all the way to 10th mil of filtrate. As you can clearly see on the left-hand panel, for the first ml of filtrate, using a nylon syringe filter, the drug recovery that is obtained is somewhere in the range of 60 to 95%. All these formulations are immediate release formulations, which means after the dissolution test is completed, quantitative recovery of the analyte should be obtained. But you can see that for nylon syringe filter, for first ml of filtrate, a significant amount of API binds to the syringe filter, leading to low recovery of analyte. On the other hand, if you look at recovery obtained after hydrophilic PTFE syringe filters, you can get quantitative recovery even for the first ml of filtrate. When we saw these results with our millipore sigma nylon syringe filters, we were concerned. So we started looking at the nylon membrane from various vendors. And we are showing data here for analyte binding for ranitidine and acetaminophen. You can clearly see that even for nylon membranes from different vendors, you can see the same binding behavior similar to what we saw in the previous slide. So nylon membrane in general tends to show higher analyte binding compared with other membranes. Nylon membranes, due to their physico-chemical nature, show strong binding to different drug molecules, and this binding will vary depending on the nature of analyte. Since a large proportion of drugs are basic in nature, this means that nylon will show stronger binding to basic drugs. What it means is for use in dissolution is that when we are trying to use nylon syringe filter for post-dissolution testing, it is advisable to discard larger volume of filtrate before the sample can be collected for analysis, because it is easy to reduce level of analyte binding once the filter is completely saturated. Here is another example of drug binding to various syringe filters where the formulation contains multiple components. This particular formulation contained 3 different APIs of interest: acetaminophen, acetyl salicylic acid or aspirin, and caffeine. As you can clearly see on this slide, when it comes to acetaminophen recovery followed by dissolution testing of this formulation, for first ml of filtrate, the hydrophilic PTFE syringe filter, that is Milex LCR, and hydrophilic PVDF syringe filter show quantitative recovery. Whereas with the nylon syringe filter, for the first ml of filtrate, you only get about 58% of recovery. For the other 2 components from the same formulation, acetyl salicylic acid or aspirin again shows very low recovery due to high analyte binding of aspirin to nylon membrane. Compared with that, for caffeine, all the 3 membrane filters, the PTFE, the nylon, as well as PVDF, does not show any analyte binding even for the first ml of filtrate. So it's important to understand the physico-chemical characteristics of your analyte as well as the dissolution medium in which the analyte needs to be dissolved, because sometimes even the dissolution medium can change the analyte binding to the membrane. And nylon is more prone to this dissolution behavior and analyte binding behavior. The next important point to consider is extractables. Extractables are contaminants that elute out from a syringe filter when you filter a sample through that filter, and they get into your sample that is used for downstream analysis. Here we are showing extractable analysis from housing material that is commonly used in syringe filters. On the left-hand side, I'm showing HPLC UV extracts from an HDPE and polypropylene housing. It clearly shows that there are no extractables seen for both the HDPE as well as polypropylene housing. Generally, very low levels of extractables from housing materials selected for Milex filter indicates high chemical compatibility of both the HDPE and polypropylene materials. In the market, there are a number of HPLC certified syringe filters. But these syringe filters are not tested exactly the same way, and they may be tested under different HPLC analysis conditions. So we looked at various HPLC certified syringe filters from different vendors. The hydrophilic PTFE syringe filters are from Millipore Sigma, the polypropylene syringe filters from vendor B, and the nylon from a vendor C. These syringe filter extractable analysis was carried out by filtration of 70% acetonitrile and 30% water through these syringe filters, collecting this extract and analyzing it using reverse phase chromatography. We used a 0 to 100% acetonitrile gradient to make sure that all the analytes that were extracted out from this syringe filter will elute under these conditions. As you can clearly see, the hydrophilic PTFE syringe filters have the least amount of extractables as seen by the cleanest chromatogram, whereas the polypropylene syringe filter show a very broad peak somewhere in the range of 5 to 7 minutes within this gradient, and the nylon syringe filters shows multiple extractable peaks within the same retention time window. So even though HPLC certified syringe filters are available in the market from various vendors, they may not be equally clean, and they may be tested under different conditions, which may be different than actual experimental work that you might be doing. So it is important to understand the difference between various HPLC certified syringe filters when it comes to using those for dissolution and the downstream HPLC analysis. Here is an example of extractable analysis that was done using a nylon syringe filter using 0.01 normal HCl as the dissolution medium or as the filtration medium. The red trace indicates that there are a high level of extractables which will not be suitable for downstream HPLC analysis. But if your method calls for using nylon syringe filter, pre-rinsing this syringe filter may be an answer. For example, if you can see the green chromatographic trace, which shows the extract that is collected after discarding the first ml of filtrate, shows much lower level of extractables that are eluting out from the syringe filter. We saw earlier that nylon also tends to show higher level of analyte binding for different analytes. And now here we are showing that nylon also tends to have higher level of extractables. Because of that, especially when using nylon membrane during drug dissolution testing, it is prudent to discard 5 to 7 ml of sample before collecting sample for analysis. Especially with dissolution testing, where the typical sample volume collected at any time point is generally 10 to 15 ml, this should not be a big problem. But it is generally preferred if we can use a hydrophilic PTFE membrane, which has very, very low levels of extractables to begin with. LC-MS is not very commonly used in dissolution testing. But if LC-MS was to be used for any other analysis like content or assay or stability, extractables coming out in mass spec are also equally important. Many of us have become frustrated by polyethylene glycol extractables that can cause havoc in LC-MS analysis. Often filters can be blamed for leaching contaminants into the sample and ruining the analysis. Here, I'm showing extractable analysis performed on filters from 3 different vendors. 8 common LC-MS solvents were used for the study of extractables from HPLC certified syringe filters. As you can see, filters with PTFE, the hydrophilic PTFE membrane, polypropylene and nylon membranes were analyzed. Multiple lots of each syringe filter were tested for consistency across the lots. Extractable tests were tested by direct infusion in ESI positive ion mode. At our site, we worked with SciEx triple quad and a Waters QTOF, and the SciEx instrument was used to obtain this data. Anyways, looking at the results, the hydrophilic PTFE membrane shows the most consistent behavior in terms of extractables from all the 8 solvents. They also showed higher lot-to-lot consistency as well as generally low levels of extractables. The types of extractables shown by hydrophilic PTFE-based syringe filters were the low molecular weight discrete molecular peaks, as can be seen in this slide. On the left-hand side, I'm showing mass trace from extracts from hydrophilic PTFE syringe filters, whereas on the right-hand side, you can see the polypropylene syringe filter from vendor B. And you can clearly see that there are large amount of peaks or extractable peaks that can be seen in this mass trace, which can become complicated during the downstream LC-MS analysis. Earlier, in increasing throughput, we also talked about using Amicon Ultra or ultrafiltration membrane-based devices for filtration of viscous solutions. It is also important to understand what is the extractable level coming from these different membranes. So comparing Amicon Ultra, which contains the regenerated cellulose membrane, with some of the other membranes from competitors like polyether sulfone, you can clearly see that the regenerated cellulose membrane has much lower levels of extractable, which makes this sample very suitable for HPLC analysis. Sometimes the extractables are not only presented from syringe filters, but even the syringes that we use for filtration of sample can contribute to extractables in the sample. This is an example from one of our customers where zinc contamination could be easily observed from the syringes itself that the customer was using. In essence, any surface that comes in contact with the sample has potential to introduce extractables as well as contribute to analyte binding. So in summary, filtration is a very simple sample preparation technique that is commonly used for dissolution testing, but it is very little understood. In this seminar, hopefully I was able to tell you about the hydrophilic PTFE membrane, which provides very broad chemical compatibility, as well as low extractables and drug binding. Followed by that, hydrophilic PVDF membrane is compatible with most of the aqueous solutions which are commonly used in dissolution testing, also has low extractables and drug binding, and it is recommended for protein analytes. So whenever you are starting a new method of analysis in dissolution testing, starting with hydrophilic PTFE or hydrophilic PVDF membrane would be your best option. Finally, selecting filter based on the throughput needs is critical because that will lead to faster or easier filtration. For example, select appropriate filter based on the sample volume, samplicity or vacuum filtration system for multiple samples, devices with pre-filters for particle-laden samples, ultrafiltration devices for removal of dissolved polymeric impurities, and automation-compatible filters for filter changing stations. With this, I would like to end the seminar, and I would like to thank all of the attendees for attending this seminar. If there are any questions at this point, I would be more than happy to take them. Thank you.
Well, thank you very much. That was a great presentation with lots of useful information. And I would just like to ask to remind the audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of the presentation window. And I'll just go on to the first question. And the first question is, in the recovery charts, is it possible to recover more than 100%? Normally the upper limit is 110% if it's declared. Could you comment on that?
Sure, absolutely. So in the experiments where you saw that the recovery was greater than 100%, this recovery was based off of labeled recovery or labeled concentration of the drug in that formulation. Whereas individual tablets might have slightly different concentration or different amount of drug in that particular tablet. And that's why you might have seen recoveries which were slightly higher than the labeled concentration.
Okay, thank you very much. And the next question is, you mentioned the Whatman number 1 paper must be taken into account in the evaluation of the filtration. And could you comment on that?
Sure. So Whatman number 1 filter paper has a pore size somewhere in the range of 8 to 15 micron. So it has a pore size similar to the Frit or cannula filters. So if you are going to use a Whatman number 1 post-dissolution testing, then you have to make sure that it is not allowing smaller particles to go through and the sample is suitable for your downstream analysis. So depending on the type of analysis that you're going to do downstream, you may be able to use a Whatman number 1 syringe filter.
Thank you. In a multi-component formulation, why do different APIs show different levels of binding to the nylon membrane?
So nylon membrane is a polyamide, meaning that it has an amino group on one end and a carboxylic acid on the other end. So both those groups have a tendency to form either electrostatic bonding with APIs as well as forming hydrogen bonds with the API. Similarly, the nylon membrane also contains the amide linkages, which are also very strong hydrogen bond formers. With our APIs having different physico-chemical characteristics, they will show differences in binding characteristics to the same nylon membrane under same dissolution condition. In the multi-component formulation example that was provided in this presentation, both acetaminophen and acetyl salicylic acid or aspirin are either basic or acidic in nature, and that's why they tend to show higher analyte binding, whereas caffeine is a neutral molecule and tends to not show any analyte binding to nylon syringe filter or nylon membrane.
Thank you. And the next question is, when can I use a syringe filter containing pre-filter versus a filter containing ultrafiltration membrane following dissolution testing?
So a syringe filter containing pre-filter is typically used when your solution contains high amount of particles, because what happens is if you have large amount of particles in your syringe in your solution, a membrane-only filter can clog very quickly, thereby preventing you to get certain volume of sample filtered. So using a high particle filtration system or using a syringe filter which contains a pre-filter will help you in those situations. On the other hand, there are certain formulations which contain polymeric excipients. And those excipients, when they dissolve in the dissolution media as you continue with your dissolution test, will lead to increase in viscosity of the medium. So these media or these solutions may not have large amount of particles, but they will contain a polymeric excipient, because of which the viscosity of solution has increased. For such solutions, you need to use an ultrafiltration membrane-based devices such as Amicon Ultra, which allows for separation of molecules based on their molecular weight rather than based on the particle size. So these filters will be able to separate out the low molecular weight API from the high molecular weight polymeric excipient, thereby providing the clear solution that is required for downstream HPLC analysis.
Thank you. And what parameters might cause a different filter to be needed for a UHPLC method versus a HPLC method?
So typically it is For UHPLC applications, most of the manufacturers recommend that filtration using a 0.2 micron syringe filter for sample is what is considered ideal, whereas filtration using a 0.45 micron membrane is suitable or sufficient for HPLC application. And again, this difference is based on the particle size of the particles present in the column, as well as the inlet and outlet frits that are present in these columns, as well as the tubing dimensions. So generally, UPLC or UHPLC use 0.2 micron syringe filter. For HPLC, use 0.45 micron syringe filter for sample filtration.
Thank you again. And the next question is, for non-protein high viscosity solutions, how long is usually needed to spin when using an Amicron ultra filter? 45 minutes seems too long for dissolution profile sample solutions. Could you comment on that?
So depending on the molecular weight of the polymer and the concentration of your polymer that is present in the solution, as well as the amount of filtrate that is necessary for the downstream analysis, the filtration time might vary. So if you look at the Amicron Ultra guides, user guides on the website, the references are generally provided, or the time requirement is generally provided for samples containing proteins, whereas with samples containing polymeric excipients, the time required may be different or may be smaller than 45 minutes. But there would need to be some kind of experimental trial and error or optimization before you can come up with a time that is needed with ultra filters.
Well, thank you. And why does analyte binding to membranes vary with analyte concentration?
So a syringe filter or a membrane filter has a certain specific surface area, and the membrane itself has physico-chemical characteristics or functional groups that affect the binding characteristics of this particular membrane. And those functional groups are fixed in a certain specific surface area of a membrane. So when you have a certain concentration of analyte present in your solution, a certain amount of this, certain percentage of this analyte will bind to your membrane or the specific surface area before the membrane surface gets fully saturated with your analyte, and no further analyte binding is observed. If your initial concentration of analyte in your starting solution is lower, then what it means is it needs higher amount or higher percentage of your analyte would need to bind before you can see full saturation of your membrane surface. So what this means in practical terms is, say for example, you are working with concentration of 10 ppm versus 2 ppm for your analytes of interest. What you might need to do is you might have to discard larger volume of the solution before you collect sample for analysis when your starting concentration is low, because that's what helps the filter to be fully saturated.
Thank you. And why do HPLC certified syringe filters from different vendors show different levels of extractables?
So HPLC certified syringe filters by different vendors are certified using different test methods. So there is no standard protocol for certifying HPLC syringe filters. And those also might be certified for extractables using different solvents. So what we did within our own study was we used a same solvent, which was 70% acetonitrile in water, for extractable testing for all the HPLC certified syringe filters that were available on the market. And as you can clearly see, they're not all exactly the same. And the reason why we chose 70% acetonitrile water as the extraction solvent is because acetonitrile water combination is very commonly used in reverse phase chromatography, which is the most common way of analyzing for doing HPLC analysis.
Thank you again. And why does nylon membrane show higher analyte binding?
I think part of this question was already answered in earlier segment, but just to repeat, nylon membrane is a polyamide membrane containing a carboxylic acid on one end and an amino group on the other end, as well as containing the amide bonds, which tend to show higher level of hydrogen bonding and electrostatic bonding. Many of our APIs are basic molecules, meaning they have higher pKa's, and they can also be charged depending on the conditions under which the dissolution test is carried out, and hence your APIs can tend to show higher amount of binding to nylon membrane. On the other hand, the membranes such as hydrophilic PTFE or PVDF, they do not contain such functional groups which will show strong hydrogen or electrostatic bonding, and because of that, they typically tend to show much lower level of analyte binding. And as you saw in the presentation, the hydrophilic PTFE membrane does not show analyte binding even for the first ml of filtrate, whereas the nylon tends to show much higher level of analyte binding.
Okay, thank you very much, Dr. Joshi, for answering those questions and for a great presentation. But unfortunately, we're out of time. I want to thank the audience for attending and participating in today's event. I would also like to thank our sponsor, Merck KGaA in Darmstadt, Germany, for making today's educational webcast possible. You will receive an email alerting you when this webcast will be available for replay. We invite you to send this announcement to your colleagues who may have missed today's live event. Thank you very much for joining us, and we hope to see you here again. All the best. Bye.